Hi, this is Ms. Lozier, and this is the Metabolism Sample Question Review. These are the short sample questions from the PDF that you can find on the website that is by Ms. Foglia. So we're going to go ahead and go through these. There's only 10 here. This shouldn't take that long. The carbon that makes up organic molecules in plants is derived directly from, so organic molecules in plants means sugars. So if I'm making sugars in plants, we're talking about photosynthesis. And it's asking, where is the carbon from photosynthesis coming from? Now, you should all remember that that carbon is going to be C6H12O6. Those are the organic molecules, our sugars. So where does this carbon come from? And I think you all know that it comes from CO2, carbon dioxide, and it's going to be the carbon dioxide that is going to be fixed in photosynthesis. And you should all remember that carbon dioxide is fixed during the Calvin cycle, and typically that is going to be, in most photosynthesis, that's going to be by the enzyme rubisco. If plants are grown for several days in an atmosphere containing carbon-14 labeled, radioactively labeled carbon dioxide in place of regular carbon dioxide, one would expect to find what? So basically this is asking us this. This carbon-14, that means that's a radioactive isotope. This is regular carbon. So if I radioactively label carbon dioxide, where would I expect to find radioactive carbon? So we need to look at our equation. It's CO2 plus H2O, and it's 6 and 6, but we're not going to worry about balancing right now. C6H12O6 plus oxygen. Now the one thing that I would hope that you remember is that the oxygen from photosynthesis is coming from the splitting of water during the light reaction. So if this oxygen is coming there, then you should know that everything else has to come from the CO2. More importantly, if I've labeled the carbon, then this carbon has to be labeled, right? So I'm looking for something that's going to have to do with radioactively labeled sugar. So would I have very little radioactivity in the leaves? That doesn't make sense. Photosynthesis happens in the leaves. I wouldn't have little, I would have a lot. Would you have large amounts of radioactive water released from the stomates? Well, no, because I didn't label the water, and the water has no carbon. Would I have a large increase in carbon-14 in the starch? So the question there is, I would have a large increase in the carbon-14, but would that be in the starch stored in the roots? So what is starch? Starch is a polysaccharide. And you guys should remember that a polysaccharide is a complex sugar. So that one's looking promising. Would I have a decrease in the rate of carbon fixation in the guard cells? Guard cells actually open and close the stomates. They're not typically the cells that are doing your carbon fixation, so that's not correct. Would I have an increase in the activity of Ruby-P carboxylase? This is Rubisco, you guys. I would have an increase in rubisco in the photosynthetic cells. So this is saying because it's radioactive, I'm going to have an increase in carbon fixation. And that might be true because like maybe the radioactivity makes it warmer in there and speeds up the reaction rate, but that's a long shot. It doesn't really have to do with the question, so my answer is going to be C. So let's go ahead and clear that off and let's move on to the next questions. So number 3 and 4. The oxygen released during photosynthesis comes from, well, we don't need to spend a lot of time on that. We just did it. The oxygen comes from the splitting of water. The end products of the light-dependent reactions of photosynthesis. So this is asking us about photosystem 1 and photosystem 2. And remember, photosystem 2 was the one on the left in our quad, and this one is going to produce oxygen and ATP. And photosystem 1 is going to produce NADPH. So I'm looking for ATP, oxygen, and NADPH, not ADP, not G3P and RuBP, not carbon dioxide, not G3P, not carbon dioxide, not protons, so that leaves us with D. That's our answer. Which of the following is an important difference between the light-dependent and the light-independent reactions? This is your Calvin cycle. reactions of photosynthesis. The light-dependent reactions occur only during the day. True. The light-independent reactions occur only at night. False. The light-dependent reactions occur in the cytoplasm. False. They occur on the thylakoid membrane. And the light-independent reactions occur in chloroplast. Yeah, that's true. They occur in the stroma, which is inside the chloroplast, but it doesn't matter because that's false. The light-dependent reactions utilize carbon dioxide and water. Now, we just said on the previous question that the light-dependent reactions don't use carbon dioxide. They do use water. They use water, sunlight, and you have to have an ADP and an NADP plus, but those get recycled. But they don't require carbon dioxide. And the light-independent reactions produce carbon dioxide and water. And the light-independent reactions don't make carbon dioxide. They make glucose. Actually, technically, if you want to be really specific, the Calvin cycle itself makes the P-gals, and then the P-gals are then made into the glucose. But glucose is good. The light-independent reactions depend on the presence of both photosystems 1 and 2. The light-independent reaction requires only photosystem 1. Okay, so let's take a look at that. The light-independent reaction requires only photosystem 1. So that this is saying that the Calvin cycle requires only the products of photosystem 1. So what does photosystem 1 make? Photosystem 1 makes NADPH, and photosystem 2 makes ATP. Yes, this also makes oxygen, but we don't care because that's a byproduct. So in the Calvin cycle, what do you have to have? Do you have to have both of those? So that's kind of a hard question to think about, so we're going to skip it. We're going to go on to E, and we're going to see what's going on with that. The light-dependent reactions produce ATP and NADPH, and the light-independent reactions use the stored ATP and the NADPH. Well, I know that that one's true, so I'm just going to go ahead and skip thinking about D and circle E. Which of the following enzymes is responsible for carbon dioxide fixation in C3? That means normal, regular old plants. And the answer is going to be Ruby P carboxylase, which I will write out, ribulose, bisphosphate, carboxylase. So, Ru, B, P, carboxylate, Rubisco. Okay, and that's the answer. So, next question. During respiration, most ATP is formed as a direct result of the net movement of, well, I'm making ATP, I'm going to have the ATP going through the ATP synthase. So I have my ATP synthase, and that's got to be in a membrane, right? So I've got my ATP synthase protein in a membrane, and it's going to go ahead and spit out the ATP. What did you have to have in order for that to happen? You have to have a bunch of hydrogen ions, and that's going to go ahead and going to provide the proton motive force is what it's called, which basically just means there's a whole bunch of hydrogen ions on one side, and so you've got a concentration gradient, and that's going to give you the energy chemically to go ahead and power your ATP synthase and make an ATP. And so it's going to be protons down a concentration gradient. That's going to be the answer. You can also see this one written a lot of times as chemiosmosis, because that is the definition of chemiosmosis. Now, that happens in both the electron transport chain in cellular respiration and in the electron transport systems between photosystem 1 and photosystem 2 to generate the ATP and the NADPH. Which of the following, in photosynthesis, yeah. Which of the following processes is carried out more effectively by a C4 plant than by a C3 plant? So C4 is one of the weird ones, right? We did C4 and CAM. Is it the light absorption, the chemiosmotic coupling that has to do with something with ATP, Tom McFadden says that, the chemiosmotic coupling in the electron to the next one. Photolysis, photo means light. Lysis means breaking, so that means I'm breaking something down with light. Fixation of carbon dioxide and transport of sugar. Now, if you remember from our quad in class, the C4 and the CAM photosynthesis plants go ahead and use an alternate means of carbon fixation. So instead of going ahead and using the rubisco, which is so important, they're going to go ahead and fix the carbon dioxide to a different organic molecule that is separated from the oxygen in the plant, either spatially in a C4 plant or temporally, a different time of day in a CAM plant. And so that's going to prevent the plant from basically eating the sugar as it makes it itself. So the last couple questions, 9 and 10. On a sunny day, the closing of the stomata in plant leaves results in, so if it's really sunny, I'm closing my stomates to save water. So am I going to, the closing of stomata in plant leaves, so my leaves are closed up. So is that going to result in a decrease in carbon dioxide intake? Yes, I will have less CO2 going into the plant. So like if here's my leaf, right? And I'm trying to get in with the CO2, and normally you've got oxygen coming out and water coming out. But if they're closed, then I'm not going to have any of that happening. A shift from C3 to C4, that doesn't happen. That's an evolutionary pathway. You are C3 or you're C4. There's no, I'm going to swap. An increase in transpiration. Transpiration is the water leaving. Okay, so you can't increase transpiration if the stomates are closed. An increase in the concentration of CO2 in the mesophyll cells. So I can't get any CO2 into the leaf, so I can't increase the concentration. Any carbon dioxide that is actually present in the leaf when the stomates close is going to be used up rather quickly. And is it going to result in an increase in the rate of production of starch? Starch is essentially, for this unit, sugars. Can I increase the rate that I make sugar if I don't have any carbon dioxide? No carbon dioxide equals no photosynthesis equals no sugar. So my answer is A. Oxygen consumption can be used as a measure of metabolic rate because oxygen is necessary for cellular respiration, right? So is it necessary for ATP synthesis by oxidative phosphorylation? This is oxyphos, which we also know as the electron transport chain, right? So this is electron to the next one. Necessary to replenish glycogen levels. That is a storage. That's a storage molecule. That's how we store carbohydrates in animals. Necessary for fermentation. Oxygen is not necessary for fermentation. Required by all living things? No, because there's things that ferment that don't require oxygen. Required to break down the ethanol that is produced in muscles? You don't make ethanol in your muscles. You make lactic acid in your muscles. Otherwise, professional athletes would go ahead and work out, and as they worked out anaerobically, their muscles would produce alcohol, and then they would all get drunk and basically just pass out on the football field, which would be funny, but not very entertaining from a competitive standpoint, and a lot of them would get alcohol poisoning and die. So my answer here has to be necessary for ATP synthesis by oxidative phosphorylation. So I'm going to go ahead and do the other ones in a different recording. So go ahead and move on to the next recording.